So hello everyone. In the previous discussions we were discussing about the types of the epithelium tissues. We have discussed about the cuboidal epithelium as well as we have discussed about the simple squamous epithelium. Today we are discussing with another very important topic and part of the epithelium tissues, that is the simple columnar epithelium. So before we begin on that, let's first discuss what is the cilia and stereocilia, because they are the modifications of some other types of the epithelium tissues. So let's begin with them. So cilia. So ciliated epithelium, or in any kind of the epithelium, ciliated means the presence of cilia. And the stereocilia is also a presence of the cilia, but they both are different. Actually, cilia can be categorized into the two parts, that is the kinocilia and another part is that stereocilia. There's a basic differences present in that. Let's discuss one of them. Kinocilia have the skeleton body of microtubule, and stereocila's skeleton body is actin filament. So this creates the difference between their skeleton body. There is another very important difference, is that they are motile or contractile, and they are not. Students, there is a basic differences present in them, that this type of kinocilia have the basal body present. This basal body generate the ATP, which moves them, or they makes them the contractile, whereas stereocilia doesn't the basal body to generate the ATP or the adenosine triphosphate. So these are the basic differences present in them. Let's move towards the type of the simple columnar epithelium. So simple columnar epithelium, as we know that their cells are pillar-like, their body is the tall and slender. So their cells are arranged in a way that they appears to be the slender cells, and they are like the pillars in view. Next thing is that they have the elongated nucleus located at the base. So we can draw an image of them. So this is something look like that. We will draw it here. So they something look like that. They have a tall cells located with the nucleus at the bases. Let's discuss something about their functions. So their function is mainly about the absorption and secretion. Let's discuss about the modifications of them. Modifications is basically on the apical surfaces. Apical surfaces may be generated with the cilia, microvilli, or the another important like flagella, anything. So first modification we have is breast bordered. In the breast bordered epithelium or modification, we have the presence of microvilli. Students, as we know, microvilli is the protoplasmic projection of the outer membrane, and it looks something like that. The basic thing is that they are non-motile. They only helps in the role of absorption. So in the breast bordered epithelium, they actually performs the role of absorption in gallbladder. Well, they help to concentrate something. Next thing is that the glandular epithelium. Glandular epithelium is basically the in this type of epithelium, or the simple columnar epithelium, there is basically a presence of goblet cells. Goblet cells are mucus producing cells. So they are arranged something like that. Let's draw the diagram of them. Is that they are located like that. This is the simple columnar, and these cells are located something like that. So they are goblet cells. Next classification of them we have is the glandular breast border. So there is a presence of the goblet cells which secrete mucus, as well as there is a presence of the microvilli on the apical surfaces of the simple columnar epithelium. So their location is the duodenum and ileum. Next we have two, that is the ciliated and the stereociliated. As we have mentioned above or the before that the cilia is the presence for the contractility and the movement. So cilia are present in the fallopian tube, the female reproductive part, and the ependymal. Ependymal is basically present in the brain to conduct the cerebrospinal fluid. These are the very two important examples by the point of view of 11th exam as well as for the point of view of NEET. Next we have the presence of the stereocilia. So stereocilia are basically the conical in shape and they are non-motile basically. So ciliated means the thread-like appearances on there, whereas the stereocilia means the conical-like appearance. So they are stereociliated. So they are actually the stereociliated cells. They are present in the vasa deferens, the male reproductive part, as well as the epididymis. So I think you have understood each and every part of the section easily. So we will meet in the next discussion with a very new topic. Till then, happy learning. Keep learning with us. Thank you everyone.